Hello and welcome to another episode of Momento Malum today. The continued improvement of any crop species relies on the use of genetically diverse parents to generate a population of individual lines in which there's enough variation to find the new improved line suited to be the next big thing. The narrow genetic base of the North American soybean breeding program across the North America region is well documented, with 35 ancestral lines accounting for 95% of the genes found in modern cultivars. The number is further reduced in the Canadian environment. For its genetic base, 5 cultivars act as a source of 65% of all the genes in the population's gene pool. These are Lincoln at 24%, Mandarin at 17%, Richland at 11%, AK Harrow at 7%, and Mukaden at 5%. One of the major cultivars of Canada is OAC Bayfield. Due to the success of this cultivar, after it's been bred in the mid-80s, it has since become a major founder cultivar of many of the cultivars being bred from it in modern times. The pedigree and a portion of its descendants can be seen on screen right now. The cited study attempts to determine how much genetic diversity went into this cultivar and how much of the diversity was captured by said cultivar, and what impact the founder populations had on the breeding of OAC Bayfield and the resulting cultivar itself. And here are the results. The foundation pedigree of this cultivar encompasses the same pedigree found in the Canadian gene pool. Specifically, Mandarin 2, AK Harrow, Richland, and Lincoln 2 are found on both sides of this cultivar's pedigree. The addition of Harrow soy reduced the genetic diversity even further, as this cultivar contains a small subset of the genes found in Mandarin 2 and AK Harrow. These genes are found on both sides of OAC Bayfield's pedigree. Another major line found within this cultivar's pedigree is the cultivar CNS. This cultivar is found in the pedigree of most North American cultivars. This is because it contains the genetics needed to resist the disease, bacterial pustule. The biggest source of variation comes from the incorporation of the cultivar Fiskeby 5. This cultivar was used because at the time, it was the most northernly adapted cultivar of soybean. This cultivar was bred in Sweden from the cultivars and experimental lines bred in Russia, Sweden, and the Ukraine. These cultivars were bred from a different genetic base than those of the North American cultivars. Much of Fiskeby 5's genetic variation was incorporated into the Canadian cultivar Bicentennial, one of the 2 parental lines used to breed OAC Bayfield. Based on the genetic analysis, it was concluded that the major source of genetic variation found in OAC Bayfield stems from Fiskeby 5. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.